Hi everyone. So in our third video on prokaryotic cells and eukaryotic cells and their similarities and their differences so that you can do your activity, which is doing a tracing and dichotomous chart for your activity 3, let's dig into the commonalities between prokaryotic cells without a nucleus in the domain bacteria and the domain archaea, and with eukaryotic cells with a nucleus in the domain eukarya. We've already talked about ribosomes and RNA. We've already talked about DNA that they both have in common. We've already talked about the plasma membrane that they have in common. Across these two domains, there's something else that they have in common. Across all of these domains, there are kingdoms in there that have cell walls, and bacteria generally has a cell wall. The grandfather of everything generally has a cell wall. Archaea mostly has cell walls in their cells. Eukarya, many have cell walls. And let's go into what a cell wall is and what it provides structurally and functionally for the cell. We worked on this a bit in our last labs, but we're going to dig further now, and it's going to have decided impacts on infectious disease and in the bacteria that you know very well. So a cell wall provides rigidity and protects the cell, especially the DNA, from the environment around it. So for bacteria, cell walls are very important to protect the bacteria. And the cell walls in bacteria are quite sophisticated. And that sophistication was lost as the cell membranes got more sophisticated in the archaea with the isoprene groups, and as the nuclear membrane developed in the eukaryotic cells to protect the DNA. But way back in the day, bacteria really used cell walls specifically with quite a bit of sophistication to try to protect the cytoplasm and their DNA. So who has a cell wall now for defense and for rigidity? Most in bacteria still, most in archaea still, and in the domain eukarya, the two kingdoms, I should correct that slide, the kingdom plantae and the kingdom fungi still have cell walls, and some protists, which we don't know what kingdom they're in, they also have cell walls as well. And when we look at the plant cell wall structure in the domain eukarya, that plant cell wall structure has a beautiful design to it, and I want you to pay attention to the glycan, which is down here. Glycan is related to sugars, and it is this rod here, this green rod. You see green rods in here. They can cross-link in all different directions, and those thin green rods add a lot of rigidity in all sorts of narrow spots. So while you are probably noticing the very big, thick cellulose microfibrils, these glycans are much smaller and can add rigidity in areas between the cellulose microfibrils. So I want you to pay attention to those glycan rods for the rather sophisticated plant cell wall structure in those higher level eukaryotic cells. Keep that in your head, because we're going to go back to the bacteria where they all came from. And we are going to really head into the bacteria's sophistication in their cell wall and their gram-positive and gram-negative cell walls, which even today stump some of the most, they stump some of our best infectious disease doctors still. So in terms of gram-positive bacteria are generally in two groups, gram-positive and gram-negative, and it provides a really great starting point for deciding what your unknown bacteria is. We can divide it into one of two camps to start with, and then we can keep dividing down till we get to our absolute identification, which is part of what you're doing when you're using these dichotomous trees, which are like biological flow charts. So this is one of the top things for prokaryotes in domain bacteria, is to find out, are you a gram-positive or a gram-negative? Well, what does that mean? Well, what you know is that, let's start with what you know. You know Staphylococcus, you've heard of Streptococcus, you've heard of Bacillus. These are known as gram-positive bacteria, and they have structurally, and then we'll get to why they're gram-positives, structurally, they have a cell wall that is very thick, and it is made up of a glycan layer that's attached to peptides. So it is called a thick peptidoglycan layer. Remember, that glycan was related to sugar. It is bonded to some peptide groups for the peptidoglycan layer. Those peptidosides are the fatty acids in the layer, and there are also sugars in the layer which are related to the glycans. This very thick layer in here is good for defense, it's good for shape of the bacteria, and it's good for regulating transport in addition to the cell membrane that is below it. So it is often a capsule, as you can see, around the bacteria. Why is it called a gram-positive? We'll get there in a second. On the other side, the gram-negatives are bacteria that you know as well. They are E. coli, Haemophilus influenza, and gonorrhea. They have a thin peptidoglycan layer right in here, much thinner. But look at what they have above, a very sophisticated outer membrane that it looks very similar to the inner membrane, but it's a little bit more complex. We'll learn about that in a second. It has additional sugars and proteins in with the lipid fat layer. And you begin to see them in there. They're more more random in here, and also the outer surface is more random. There's some thin areas and some thick areas. There are fibrils outside of the surface. This is a very differentiated outer membrane, and as a result, it produces a very tough target for infectious disease physicians. We'll explore that in a second. When we talk about gram staining, which was only known since the late 1800s, staining is a decisive way to divide your unknown bacteria into one of two camps. Either the bacteria absorbs the water-soluble gram stain material, which is called violet. It's a special, very small molecule, easy diffusion, and water soluble, that is absorbed by the peptidoglycan layer, because sugar is going to absorb a water, so that this gram stain is taken in by this thick peptidoglycan layer, and in doing so, it shows the violet stain on here. This is a representation. This is a microscope slide of gram-positive and gram-negative stains together. The gram-positive stains are looking violet with the water-soluble gram stain. And generally, when we see this water-soluble stain taken up with microbes, generally, very generally, that can mean easier antibiotic treatment, because it will absorb water-soluble materials right in through the membrane. Not always, though, and that lends itself to multiple resistance factors and mutations. So generally, gram-positive bacteria are treatable in a very general way. There certainly are gram-positive bacteria that are running rampant around our hospitals, including Staph aureus, methicillin-resistant Staph aureus. So generally, gram-positives tend to be treatable because they have that water-soluble layer. You can treat them with an infusion of antimicrobials in saline. However, gram-negatives are not going to absorb the gram stain. That gram-negative is a bacteria with an outer membrane of a phospholipid membrane, well, of a polysaccharide lipid membrane. And that polysaccharide on top, that might take in some of the gram stain, but not the lipid. And in fact, in gram staining, you wash off a lot of the extra stain, and gram negatives are just not going to have it. So these stains are not absorbed by the outer lipid membrane. And if you think about it, gram negatives as a result are going to be tougher to treat. This is a much more diverse surface to get through and predict. This is a very sophisticated membrane down here that is tough to get through. Gram negatives, E. coli, Haemophilus influenza, and gonorrhea are tough bugs to treat because it's tough to get in through the cell membrane. Now, in terms of the higher level cells, the eukaryotic cells, we were talking about, well, what's in their cell wall? Their cell wall may not have the sophistication of the bacterial cell wall because there are other layers of defense in eukaryotic cells. However, there is differentiation on the materials that are in the eukaryotic higher level cell wall. Like you notice here, in kingdom plantae, there is cellulose in the cell wall. Those are the blue rods here. In kingdom fungi, there is a material called chitin, and that might be in. In protists, which are not in a kingdom, that could be cellulose, meaning it's very much like a plant, or it could be even silica. But all of these materials are structurally important. Now, we've talked about the things that all of these cells have in common: RNA, DNA, plasma membranes. Now let's get to energy sources and cellular metabolism and cellular respiration and what they have in common between all of these cells. Cellular metabolism is the ability to take in nutrients into the cell, and that is generally through an endocytic process to move these materials into a cell. That's the case for all of these cells. They convert and metabolize glucose or their fuel into ATP. And cellular respiration takes that ATP and converts into ADP for an energy source. So cellular metabolism and respiration work together and tend to be right in the same area. For prokaryotic cells, that's simply in the cytoplasm. These cells are so small that these areas in the cell are so close to each other that the metabolism and the respiration is done simply in the cytoplasm itself. With eukaryotes and the higher level cells, that moved into organelles, which had their own membranes to them as well. And so, for instance, you might have vacuoles, which are storage spaces for food and water in these much larger eukaryotic cells. And from those vacuoles, that food could move into mitochondria, where metabolic and respiratory processes would take place to move from the nutrient source into ATP